Okay. Phage therapy by me. This is the introduction. What are phages? Life cycle of phages, color and pictures of phages and bacteria.
Criminal list.
Put a criminal list of viruses just so you can give me more interest in them generally. I also talked about amino acids and how to make a virus if you really want to make some biological warfare. If you want to use biological warfare, then use that. Okay, what are phages? Phages are just a type of virus, right? Viruses generally, right? When you see a long time ago, people usually define viruses by not so long ago, people thought that all viruses cause some sort of disease, and early definitions usually include something about disease, like, I don't know, they cause very severe diseases or maybe causing cancer. Some viruses do cause cancer. So they kill bacteria, which is the fun part. However, why is it fun when, viruses are bad, right? Are they bad? Phages aren't necessarily bad, but one thing before you note is that one type of phage can only infect one bacteria. Why is that? Because of the tail fibers. These are these little things that look like they're jutting off this thing, right? And this is a phage, a typical phage. Phages come in many shapes. They can come in balls. They can come in this shape, and I don't know. There's many more to be discovered. So you could theoretically discover a different new type of phage, which is a lot of different things. These tail fibers only stick to one cell membrane, right? And that's why these viruses can only infect one species of bacteria, maybe even some strains of bacteria. It's weird. They're used as medicine, really, because it's first weird. Viruses are not supposed to cure infections. They're supposed to cause infections. That's typical reasoning. However, they were used as medicine in the former Soviet Union. However, why former Soviet Union and not here? We don't find phages in the market because, we think that, viruses are very bad. We want to try to get rid of them. Let's just say, because phages were thought to be, we used to think we could use as a medicine, but that was in 1915 when they were discovered. And then 1940, pow, antibiotics. You've probably heard of them, and you've probably almost definitely have taken these antibiotics. Antibiotics are they're just generally chemicals. Okay, they're generally harmless and they don't do anything. Western people thought they were generally not so bad, and we were like, hey, we solved bacterial infections, and now no other virus will cause anything bad at all. But hey, oopsies. I guess I'll need to do stuff. So these phages, they live everywhere, right? These phages live in places like dirt. And I'm going to mention that they definitely will live in water that hasn't been chlorinated. And if it has, there's probably a lot of dead viruses. And they have a very wide range of genes, right? Okay. They can have around 4 to around 200 genes. I mean, the 200 I just kind of guesstimated based on how kind of my how I inferred on the pages. And they can have as much as 4 genes, which is weird because how does an organism live with just 4 genes, right? I mean, phages are still complex to me, right? This is how phages do their killing, okay? Well, they have to kill bacteria in order to reproduce. Okay, I think it's sad, but many people think that just killing bacteria is a good thing. I kind of think it's still a little sad. These phages, they get released, right, from their host, and then basically they just like go wherever, like whatever, any natural changes, they will stick to the fibers. They can also kind of walk along the cell. Well, that's the only thing, kind of walking they do, right? And they inject their DNA boom, into there. They just like squeeze their tail in. And then once they do, they just inject their DNA. I already said that. And then let's just say some viruses do this very stupid like move, right? Because this is the stupidest part of this. I don't really think it's a very smart one. But the virus basically becomes part of the host by injecting, well, the DNA becomes embedded in the host's DNA, which is weird. And then basically makes more DNA and makes more cells. And this process can be good, right? And that means the virus ends up in all of that bacteria's descendants. The phage could still, if what it wants, like this is the lytic cycle. I'm talking about the lysogenic cycle first, because it's generally more boring and more fun when the good stuff happens, which is what I'm going to say next. When when a trigger, right, when like some environmental condition, when times of stress, the phage can separate from the host and be can be another entity that's not, it's like separate from the bacteria. Well, unless there's mutations that just riddle it so it can't get out anymore. And then once it becomes a separate entity, it just makes more stuff. In like 30 minutes, in 30 minutes, it just makes more of itself in just 30 minutes. It just makes around 100 cells. Like basically, if until as like around 100 phages, new phages, and that's for one of the phages that I researched on. It's not for all phages, I guess. But in 30 minutes for that single phage, pow! All these descendant bacteria die because of these phages. And basically the same thing happens again. Very easy, right? Or there's another cycle called lytic cycle. This is one more straightforward and kind of less fun one, and kind of boring. You see, once they go out, right, they don't embed themselves in host genomes. They just then destroy their host DNA, right? They destroy it. And they force the host to make some of the proteins. Not that the virus still makes some of its own proteins. It's not like that they don't. And then they produce all the parts are needed to make new phages. And then they just produce this protein called lysine that destroys the cell walls. So this bacteria dies in this way. And they're able to get out. How much bacteria are lysed every day? They call it seems like so. Oh. So basically, 25% of the 25% of the ocean bacteria population are killed this way. Oh no. Oops. I think I have a problem. Back to that. This is how it basically injects the DNA. This is the basic color of these phages. This is a T4. This is an E. coli phage. E. coli phage. It's called T4. You'll hear it very often. And I also, you might be kind of wondering, right? You might be not saying that, but this is white, right? Which is weird. First, I did show you the colored phage, right? And this doesn't make sense. This must be like some old photograph, right? Wrong. It might be an old photograph. I'm not sure if it is or not, but this is exactly what you see if you look at T4 phage in a microscope. Most viruses don't have color like we do. There's just black and white in this picture. Same with most viruses. Color is usually made by a pigment, which is like sort of chemical that kind of makes the pigment process. It's found in our DNA. It could be anywhere, literally. Color is usually made by pigment. You know, viruses don't have any good reason to make pigments because literally, why? Like if you're a virus, what's the good reason to make pigments anyway? It's not like they can camouflage or anything else. So they just basically stay colorless and it's not very interesting to me at all. People just use color with viruses because of for interest, right? Because this looks less appealing than the one that I showed at the beginning of the slides. And because of anatomy, it showed different features. So, what can you see from this phage here? Can you see anything I talked about? Can you even see it at all? Okay, this is the legs, same. These are the whiskers. I should have said that earlier. And this is the head again, where the DNA is stored. This is the tubey body. But if you're kind of like very sad, right? You should look at those endoviruses. Most of them they look blue, and their hosts. Oh yeah, their hosts. Their hosts will look blue as well. Which, yeah, just not going to be looking good. So this is basically another one. I showed you this because phages are completely very, very small. We don't know a lot about these phages, right? So this is one of these phages on the E. coli. And you can see how many are there. I'm not sure how much are there, but I think there's like, I don't know, 1,000 of them could be, no, 200 could like cover one side of this bacteria. Literally. I'm not kidding. This is the criminal list of viruses. I just put this up for fun, right? This is a virus called a mammavirus. And its host is amoebas. This is one of the largest viruses. It's very weird because, well, it's very large, and considering that, it's even larger than some bacteria, in fact. Well, it's not the largest virus. The largest one is actually the oldest one. It's called Pictovarius cerberium. It's still very large for a virus, and it doesn't behave like other viruses. For example, when it infects a cell. It doesn't just basically put its DNA inside the nucleus like many viruses that infect animals do, right? It just basically turns itself into a vaccine. Using help with the host, of course, because viruses need host to reproduce, as host is one of the smallest organisms found in water, amoeba. And okay, so this is still the E. coli phage, right? Enterobacteriophage T4. Its host is also the E. coli genus, which is also called Enterobacteria. But I'm not gonna. Let's just call it the E. coli genus for simplicity because it's just very boring. Anyway. Oh, you'll probably heard of this. This is smallpox. Oh, this is very, very bad. I saw one of those photos on that disease, and literally, I'm very glad that you don't have the disease. If you have the disease, it'll be like boobies everywhere, like smallpox scars, like even here and here, everywhere. Like your whole skin will be hard. It's like slowly, I could not bear it for even a second. And it tells us humans, this is these smallpox. Smallpox still exists in 2 laboratories, even though it's kind of eradicated, it's not yet extinct. And in these 2 laboratories, that's where smallpox is, right? It's in there. It's under very tight security. You can't just walk in and get smallpox samples. No. And next, researchers have also figured out how to make smallpox virus for just $100,000. So, in fact, if you have that much money and you have a lab, right, you could make a smallpox virus. Well, yeah, that would be cool and a little dangerous because smallpox kills. It kills 30% of the people who get infected. This is the first virus that we knew of, tobacco mosaic virus. So this one infects tobacco. It causes the leaves to look like kind of like it has a spots, causes the spots in there. Host is the tobacco plants. It's very simple. See, look, there's just protein and RNA. That's what I mean. See, that's why you never don't buy tobacco mosaic virus. Well, why would people want to buy it anyway? But you could probably just make this tobacco mosaic virus easily. And there's this protein molecule just for the nice coat shape because RNA by itself doesn't look that appealing, right? This is the weirdest one. It's called the Adigenous two-tailed virus. It's called a two-tailed virus because it has these 2 tails. Very weird. Not usual for viruses. This two-tailed virus, once it gets out of the host, it actually kind of grows, sort of. Basically, it's first like a lemon, right? But it grows the tails and then shrinks its body, the lemon, up to 1/3 of its size. We don't know what the tails are for, actually. I don't know what it's for. No one knows it's for, but it could be used to get in the host. We could study more and we can figure out. Its host is in the archaea in the genus Adigenous. Kind of hard to say. And amino acids. Amino acids, they make proteins. Very important. They make cell walls. And